All right, so it looks like the run has started. It didn't make it up to temperature, so I moved it inside the car, but it's running right now. And what you're looking at here is what's called the channels panel. So these are all 126 of the pores, and they're color-coded to tell you what's going on there. So it turned out that if you look at the envelope here on the upper right, it says we had 82 pores that are available for sequencing out of the 88 that it came with. Which is awesome. Usually what you do is you lose a lot of those pores if your quality of your DNA is not good, really high quality. The trick to Oxford Nanopore Sequencing is super high quality DNA. That's really what you're after. I put low quality DNA in there and totally shot out the entire flow cell. It had 0 active sequencing nanopores or pores, so that can happen. So this looks good. What you're seeing there is we got 44 inactive at this time. There's some unclassified. There's some that are recovering. So they've sequenced a strand of DNA, and now they're recovering to maybe sequence another. And the numbers are kind of jumping all around there. The light green ones are the actual ones that are sequencing at that second. And this Oxford Nanopore sequencer, it'll sequence DNA around 200, 300, or 400 nucleotides a second. So very fast as that DNA strand goes through. And so it's jumping up and down how many are sequencing in the light green. Then there's some dark green, which are pores that are available for sequencing, and there's no DNA being threaded through the nanopore at that spot. So this is what it looks like, and that's what it'll keep doing here for as long as we want to run it. So eventually those pores, those inactive pores, will increase over time, and this will actually run out of actual sequencing pores here. So this is showing you some of that same kind of data, just in a different histogram type format. Similar, so we're up around 82 channel count pores that have the ability to sequence. And then we can come out over here. This is showing us the histogram of the reads that are sequenced here. So let me go to estimated bases. Here we go. So here's the first, it's actually calling them as they're coming through at this moment. Here's the first 5 sequences that went through, and it shows their size right here. So we've got one that's around 200, one are almost to 237 base pairs or nucleotides long. Then we've got 4 that are up there a little higher, right around, looks like 350, 400 nucleotides long. And it'll just keep sequencing and add to that as it goes. And we can run this thing for 8 to 16 to 20 hours. But the general idea of this Flongle or Oxford Nanopore sequencer is that you don't really need to run it that long. Usually you just want to come out, run it to get enough data to find out what is that sample. That was kind of the main purpose of this, which is to come out, identify a pest, and say, well, what kind of pest is that? What exactly is it? What's its genus? What's its species? What family is it in? Is it a known vector for certain viruses? So more you'd learn about your pest or your pathogen, then the more likely you're going to be able to treat that crop, that agriculture that you're working on. That's the general idea. So probably what I'm going to do is let this keep running here for a little bit. I might even just leave it in my car and drive home, and then by the time I get home, I should have a bunch of data, can then analyze it. There are multiple ways to analyze the data at the next point. You can do it in real time using something called Epitome, which I have on my computer. I don't have it on my cell phone. You can also use just your computer to run searches online, do blast searches, or you can use programs like Genius, which is actually what we're going to be using in the lab to analyze this data. And you can map that data against known aphids to find out what species of aphid is that, to learn a little bit more about it. Okay, so I'm going to go ahead and cut it there with that beautiful view of the channels panel. and that DNA sequencing through there.